So here is glycolysis. This is the 10-step pathway in producing glucose into two molecules. So I want to reiterate, you're going to make two molecules of pyruvate, 10 steps, 10 enzymes. I want to bring your attention here is that some enzymes go in one direction, such as the first enzyme, that's hexokinase, goes in one direction. All right, our second enzyme that goes in one direction is this big important enzyme PFK, phosphofructokinase. And our third enzyme that goes in one direction is the last terminal enzyme in glycolysis, pyruvate kinase, and that converts phosphoenol pyruvate to pyruvate. So three enzymes that go in one direction. The other reactions are in an equilibrium, so they can go back and forth. The reason why I'm telling you this is that we will be studying glucose being converted to pyruvate. So I can ask you guys the question, how will we study the reverse, gluconeogenesis, the building of glucose? How will we study that? That is, how will we study pyruvate going all the way back up to glucose? Well, I want to convey to you that it's easy. All you have to do is go in the reverse of all these steps except at three enzymes. So what are those three enzymes? This enzyme. So this is actually two enzymes that can go in the reverse direction. But it's going to be an enzyme here, a unique enzyme for gluconeogenesis, a unique enzyme here for gluconeogenesis, and then finally a unique enzyme here for gluconeogenesis. So we could study glycolysis, but I can also teach you gluconeogenesis. The only thing I would have to teach you are the enzymes that are going to reverse this step. Okay. The enzyme that will reverse this step here, phosphofructokinase, and then the enzyme that is going to reverse this step here, hexokinase. So you know glycolysis. All I need to do is teach you three, it's actually four, but because this last step is two steps, but I can teach you gluconeogenesis just like this. All I got to do is teach you four different enzymes, four separate enzymes. Everything else is the same. But for now, we're just going to go downstream. Okay, glucose to pyruvate, and if you're more interested in the reverse stage, that's gluconeogenesis, you're going to have to know only four other new enzymes that sort of reverse the enzymes that go in one direction. All right, our final introductory slide with glycolysis is, you see, why do we take glycolysis, glucose, and forming 2-pyruvate, and why do we split it into 10 steps? And this slide actually potentially answers that. We split this up into 10 steps because we have the capability of producing some really, really energetic molecules. And those two are phosphoenol pyruvate and 1,3-bis phosphoglycerate. These two molecules have what we call high group transfer potential. So if you remove that phosphate from phosphoenol pyruvate, that phosphoenol pyruvate becomes pyruvate. It sort of lowers the energetic bar. So phosphoenol pyruvate is a very tightly wound molecule. Okay? It's bursting with energy. When you relieve that phosphate group and phosphorylate ADP to ATP, it's like releasing a burden. Okay, the energy becomes a low energy compound, but you've produced ATP, which is a wonderful compound for the cell. The same thing with 1,3-bis phosphoglycerate. When you release a phosphate here, okay, making ATP, that's about minus 50 kilojoules of free energy that is released. So that is energy that can be harnessed. Same thing with phosphoenol pyruvate, a very high energy, highly charged molecule. When it releases its phosphate to produce ATP, you release minus 62 or so kilojoules per mole. That's a lot of energy that can be captured for the cell for purposes of doing other things or for purposes of just entrapping that potential energy in the form of ATP. So splitting glycolysis or any metabolic pathway into 10 steps is very good because you produce high energy molecules or molecules with high group transfer potential. That is releasing that phosphate group really makes that molecule go from a high energy state to a low energy state. That phosphate can then go into making ATP. So very, very critical step, the production of high energy intermediaries in glycolysis. So now we will study glycolysis. The first thing we'll do is get glucose in. So usually if we're talking about the muscle cell or the liver cell, red blood cell are very important in the brain. You have to get glucose in. So glucose usually comes in via these family of transporters known as the GLUT family of transporters. So once that glucose goes in, you sort of want to sequester it into the cell, and we sequester it into the cell by converting it into glucose 6-phosphate. So we phosphorylate the number 6 carbon, and that's called glucose 6-phosphate. The enzyme that does this is called hexokinase, and automatically a pretty bad omen here because we've utilized one ATP molecule. So here I am telling you that we make ATP in glycolysis, at least 2 ATPs in glycolysis. The very first step, we actually expend an ATP molecule. So that's not a good thing, but let's see what happens when we go to the other steps. So our first step is making glucose 6-phosphate. Glucose goes to glucose 6-phosphate. The enzyme that does this is hexokinase, and we have magnesium sort of chelating and neutralizing the negative charges. So we have the liver version, which is called glucokinase. It's very specific for glucose. Hexokinase is what we call a promiscuous enzyme in the sense that hexokinase can phosphorylate glucose, but it can also phosphorylate other 6-carbon sugars, such as, for example, fructose. So the difference between hexokinase and glucokinase has to do with specificity, it has to do with Km, and it has to do with the fact that the liver version, glucokinase, has a lower Km, enhanced specificity for glucose, whereas the hexokinase version that's found elsewhere outside of the liver is more indiscriminate and more promiscuous for phosphorylating other sugars as well. So here we have our ATP molecule and glucose. We have utilized one molecule of ATP. Magnesium is very important in this reaction. Why do we need magnesium in the body is actually the reasoning is to sort of charge, neutralize the excessive negative charge of these phosphates. So that's the metabolic role of magnesium in the cell. Hexokinase is a classic example. We just learned this from the very get-go in our biochemistry class, at least when I took it 20 years ago, of this induced fit. We could also call it Pac-Man. So here is our glucose molecule and then substrate. Once it gets into the active site, the enzyme sort of swallows it up, kind of like Pac-Man gobbling up the Pac-Man food. So a lot of things happen here. Water gets exuded out or exuded out, and it gets excluded. And the active site, significant conformational change sort of envelopes that glucose 6-phosphate molecule. So hexokinase is the first reaction of glycolysis, phosphorylating glucose to glucose 6-phosphate. The second reaction of glycolysis is a simple isomerization reaction. It's taking glucose 6-phosphate, which we know is a ketose, and converting it, which we know as an aldose, and converts it to the ketose. So this is simply an isomerization reaction. The enzyme that does this is phosphoglucose isomerase. This is reaction 2 of glycolysis. So mechanism is shown here. The idea of phosphoglucose isomerase is to actually open up the ring. So here is our glucose 6-phosphate. An acid-catalyzed ring opening occurs. And then here we have a proton that's taken up by a basic amino acid. A carbonyl group gets formed here. So here we have a double bond, sort of stabilizes the hemiacetal. The double bond goes into the carbonyl group, and then this oxygen here sort of closes in. And now you have the ketose form, and then the fructose 6-phosphate gets released. Another substrate can bind. So it's an acid-catalyzed ring opening. You have to abstract a proton in the sugar molecule, generate this carbonyl, and then nucleophilic attack on the carbonyl gives you fructose 6-phosphate. Glucose 6-phosphate and fructose 6-phosphate are isomers, so it's very easy to catalyze from the aldose form to the ketose form. It's basically an acid-base catalysis that phosphoglucose isomerase does. Phosphoglucose isomerase is linked to anemia. In fact, a lot of these enzymes that are mutated in glycolysis or defective in glycolysis give rise to anemias. Hemolytic anemias is most common. Our third enzyme is a big one. It's phosphofructokinase. So if you look at a biochemistry textbook, they will dedicate several pages to this enzyme, but I will condense it down to a couple of key points here. The key point you want to recognize about PFK, PFK1 I should tell you, because there's also PFK2, but this is PFK1 in glycolysis, and it is the regulatory checkpoint in this pathway. Regulatory checkpoint, rate determining enzyme, rate limiting enzyme, all of these are very important terms in discussing phosphofructokinase. So what does that mean? A regulatory checkpoint enzyme, the committed enzyme, rate limiting enzyme, basically means is that once fructose 6-phosphate gets converted to fructose 1,6-bisphosphate, once PFK is on, that means the cell is committed. It is committed to glycolysis. And you can actually see that in the arrows. So you notice the unidirectionality of that arrow. It means it's the committed step. Once flux through phosphofructokinase is on, okay, once flux through this protein is high, that means the cell is dedicated itself to glycolysis and glycolysis only. So we call that the committed enzyme. Lots of terms for this. Committed enzyme, rate limiting enzyme. Some textbooks also call this the bottleneck enzyme. It's the enzyme that commits. It's the enzyme that commits this to glycolysis. And, well, a second ATP molecule is used up. The first one was in hexokinase, and now we utilize a second ATP in reaction number 3. It doesn't look good in terms of ATP counts because we have a debit of 2 ATP molecules, but now we make fructose 1,6-bisphosphate. Committed step in glycolysis, and a committed enzyme is going to be heavily regulated by allosterase. It's going to be activated by AMP. So usually that means the cell is in a very low energetic state. So AMP is low. AMP will bind outside of the active site of PFK and increase flux. Get glycolysis moving because there's high AMP, low ATP. Okay, on the flip side, if there's high ATP, some of the ATP can go back and downregulate this enzyme, alter the Vmax, alter the Km. So ATP negatively regulates this enzyme. Citrate actually negatively regulates this enzyme. The reasoning behind this has to do with the citric acid cycle, separate metabolic pathway. And if the citric acid cycle flux is high, that means some of the citrate can then come out, go into the PFK allosteric site, and lower flux through glycolysis. Why? Because the citric acid cycle is on, everything is really moving along. You want to slow down the flux here. It's sort of a way to calm down the enzyme. And then this fructose 2,6-bisphosphate is an allosteric activator of PFK as well. So I'm not going to talk about fructose 2,6-bisphosphate. That's an allosteric effector that's regulated by insulin. It's regulated by glucagon, and it's beyond the level of this course. But fructose 2,6-bisphosphate is actually made by PFK2. Okay, so PFK1 does this reaction, glycolysis. PFK2 is involved in making the activator for PFK1. So that's fructose 2,6-bisphosphate. And for the biochemistry 1 students, we will not talk about that, but we definitely will talk about it in my next lectures for the advanced students. All right, so remember PFK is a rate-limiting step regulated by several allosteric effectors. Reaction 4 is catalyzed by aldolase. Okay, so we take that fructose 1,6-bisphosphate and we're going to split that molecule right here. And when we split it, we get dihydroxyacetone phosphate and we get glyceraldehyde 3-phosphate. So notice here the arrows back and forth. You want to keep an eye out for the arrows, those reactions that go one way and those reactions that are at an equilibrium. They can go back and forth. Shunting, main point here in metabolism in general. This dihydroxyacetone phosphate can be utilized to make lipids, okay, like your phospholipids, your glycerol-based phospholipids. This is a stepping stone for that. So is glyceraldehyde 3-phosphate. It's a stepping stone to make lipids. So this can be shunted elsewhere to other metabolic pathways. All right, so we split this molecule in half in reaction 4, and we get dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Right, so the enzymatic reaction here in terms of mechanism has to do with the Schiff base intermediate. So this Schiff base here is really, from an organic chemistry perspective, is a reaction of a primary amine with a carbonyl group. So here the primary amine is given by the lysine active site residue. It's going to react with the carbonyl group of fructose 1,6-bisphosphate. The Schiff base is very, very unstable. So due to the instability of the Schiff base, this leaving group becomes glyceraldehyde 3-phosphate. So that's one of your product that comes out. And then we have another, this is going to be a aspartate side chain that's going to create the final product, and the final product is dihydroxyacetone phosphate. So we have a lysine and an aspartate. Those are the two active site residues. Okay, it functions via a Schiff base intermediate. Primary amine with a carbonyl group is a Schiff base. It's very unstable, but it allows for the leaving group of glyceraldehyde 3-phosphate. Aspartate comes in the way and does some nucleophilic attack to regenerate the second product, the second product, dihydroxyacetone phosphate. So mechanisms, I'm not going to ask a lot of questions on mechanisms, but this will really help you learn and imbibe what's happening in terms of the intermediary. So, just memorizing glycolysis for memorization's sake is not going to help. But if you look at it from a mechanistic standpoint, it actually does help to see what's going on. So that's the reason why I'm going over the slides for the mechanisms. All right, so that was reaction 4. Reaction 4 was aldolase. Reaction 5 is triose phosphate isomerase. And triose phosphate isomerase actually is going to do one thing and one thing only. It's going to take the dihydroxyacetone phosphate that you made before in the previous reaction and convert that to a second molecule of glyceraldehyde 3-phosphate. So if I were to go back a couple of slides here, you see aldolase makes glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The fifth enzyme, triose phosphate isomerase, is going to convert that dihydroxyacetone phosphate to another molecule of glyceraldehyde 3-phosphate. So thanks to the fifth enzyme, triose phosphate isomerase, from here on out, you're going to have 2 glyceraldehyde 3-phosphates. 2 glyceraldehyde 3-phosphates. So triose phosphate isomerase, a couple of other trivia concerning this enzyme. We call it a catalytically perfect enzyme because it has a high kcat over km. The minute dihydroxyacetone phosphate gets into the active site, bam, almost faster than the snap of my fingers, you'll get glyceraldehyde 3-phosphate. So it's a very, very efficient enzyme. And the efficiency comes from a very high kcat over km ratio. It's about 10 to the 8 or so. Okay, very, very large number, kcat over km. So how fast this enzyme proceeds is just how fast the dihydroxyacetone phosphate gets into the active site. Once it does, the generation of your second molecule of glyceraldehyde 3-phosphate is extraordinarily fast. So we call that a catalytically perfect enzyme. Also, there is a disease related to triose phosphate isomerase, and the disease is called methylglyoxal toxicity or methylglyoxal blindness. Methylglyoxal is a very toxic metabolite that is an offshoot of glycolysis. So if I were to go back here, dihydroxyacetone phosphate, in some cases of glycolysis, sometimes it will lose that phosphate, and this alcohol gets, this hydroxyl group, I should say, this hydroxyl group gets oxidized to the aldehyde. So sometimes, not in all the time, but in very rare cases, this dihydroxyacetone phosphate can spontaneously lose that phosphate, oxidize that hydroxyl group, and produce methylglyoxal. So that's a very dangerous byproduct. We actually also see that in some types of cancers, the formation of methylglyoxal. It's just a very dangerous product that could happen stemming from dihydroxyacetone phosphate being accidentally converted to that. So triose phosphate isomerase, particularly this blue random coil region, prevents that from happening. So that random coil region sort of locks the substrate in place so that dihydroxyacetone phosphate does not become this dicarbonyl group, okay, this dicarbonyl intermediary. So this is a case where the structure, particularly this random loop, does not participate in the active site chemistry. What it does do is that it participates in preventing a formation of methylglyoxal from DHAP, dihydroxyacetone phosphate. If you take a protease and cut off this blue random coil region, you will get methylglyoxal formation, which is very bad. It's a toxic intermediary of glycolysis. Also, glycolysis halts. Okay, glycolysis stops. The random coil region sort of sets everything in place so that dihydroxyacetone phosphate does not spontaneously convert into this dicarbonyl aldehyde that we call methylglyoxal. So a little bit of a clinical relevance of that random coil region in this enzyme triose phosphate isomerase. All right, so we are halfway through glycolysis, and we have spent 2 ATPs, and we have made 2 glyceraldehyde 3-phosphates. Don't forget, the rate determining enzyme is phosphofructokinase, PFK. Okay, one direction. It takes fructose 6-phosphate, converts it to fructose 1,6-bisphosphate, a very important enzyme in the sense that it is the rate determining enzyme in glycolysis. All right, since this enzyme triose phosphate isomerase makes 2 molecules of GAP, glyceraldehyde 3-phosphate, from here on out, reactions 6, 7, 8, 9, 10, we're going to multiply everything by 2. So instead of 1 molecule of GAP going, 2 molecules of GAP are going. Okay, glyceraldehyde 3-phosphate. G3P or GAP both stand for glyceraldehyde 3-phosphate. All right, the last stage here of glycolysis, we better produce some ATP, okay, because we spent 2 molecules of ATP, and so what's going to happen here? Also want to tell you, we make ATP in glycolysis, but really the bang for your buck in making high amounts of ATP comes from 2 things, not from glycolysis, unfortunately. They come from ATP. The ATP formation comes from electron transport chain. So you can make 33 to 34 ATP molecules through the electron transport chain. Also breaking down fatty acids. Okay, if you break down a fatty acid, you can get up to 100 to 110 ATP molecules. So in terms of generating energy in the form of ATP, glycolysis is kind of puny. It's not the best way, but it's a very important way for muscle cells because you need it very fast, and it's also very, very crucial in the brain because the brain does not metabolize fatty acids to get you all those 100 or so ATPs. And so really, glycolysis is very important for muscle cells, very important in liver cells because liver is the main sensor for glucose, and it's very important in our brain. All right, back to glycolysis, reaction 6. Glyceraldehyde 3-phosphate, okay, remember we're going to multiply everything out by 2 because we have 2 molecules of this. Glyceraldehyde 3-phosphate is going to be converted to 1,3-bis-phosphoglycerate. The enzyme that does that is glyceraldehyde 3-phosphate dehydrogenase, GAPDH. It's an oxidation reduction reaction. This NAD plus gets reduced to NADH. That's a good thing. Okay, making NADH is a very, very good thing. Why is that? Because of the electron transport chain. This can donate its electrons through the electron transport chain and make a lot of ATPs. It's also a good thing because the NADH can go into other dehydrogenation reactions, right? So it's always good. In fact, the cell has a reservoir and a pool of NADHs that it could sort of capture when it needs to do a oxidation reduction reaction. Okay, so 1,3-bis-phosphoglycerate makes NADH, which I want to hopefully convince you is a good thing. And this inorganic phosphate goes into right here. Okay. Reaction is endergonic, positive value of free energy. So usually requires an impetus in terms of energy. And also we make 2 NADHs and we make 2 molecules of 1,3-bis-phosphoglycerate. Remember, everything from here on out gets multiplied by 2. So here's the mechanism for glyceraldehyde 3-phosphate dehydrogenase. Again, the mechanism is to make you learn, not so much test question material. First of all, glyceraldehyde 3-phosphate dehydrogenase is ping, 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 pong, pong. Okay, ping, 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 pong, pong. So here's our ping, glyceraldehyde 3-phosphate is our ping. Our second pong is NAD plus that comes in. Here we have a cysteine active site. And here's your NAD plus gets converted to NADH. So ping, ping, ping. And then the phosphate comes in, makes it the third ping. And the phosphate does nucleophilic attack. Pong is 1,3-bis-phosphoglycerate. Pong is NADH. So ping, 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 pong, pong. And hopefully we remember this from our inhibition lectures on competitive and non-competitive inhibition. Arsenate will uncompetitively inhibit this step. Okay, remember, arsenate causes a lot of pustules in our fingers and toes, the extremities, and arsenate specifically found in a lot of contaminated drinking water. Arsenate sort of binds as an uncompetitive inhibitor at this step, displacing the phosphate. So arsenate is an uncompetitive inhibitor of glyceraldehyde 3-phosphate dehydrogenase. So this is mechanistically what's going on. The byproduct here is 1,3-bis-phosphoglycerate. All right, reaction 7 of glycolysis is phosphoglycerate kinase. So that 1,3-bis-phosphoglycerate that we see here, high energy molecule. Okay, high energy molecule. Molecule is very, very primed. It's sort of unstable. Okay, it's brimming and brimming and brimming. It wants to get to a lower energy state. How do you get to a low energy state? You release that phosphate. When you release that phosphate, you get ATP. You actually get 2 molecules of ATP because 2 molecules are going through. And this 1,3-bis-phosphoglycerate gets converted to the less stable, highly relaxed molecule known as 3-phosphoglycerate. And we're going to call this ATP formation substrate level phosphorylation. It's not ATP that's formed through the electron transport chain. It's not ATP that's formed from fatty acid metabolism. It's ATP formed by literally phosphorylating an ADP molecule. So the enzyme that does this is phosphoglycerate kinase, coming from 1,3-bis-phosphoglycerate, and we make not 1 but 2 molecules of ATP and 2 molecules of 3-phosphoglycerate. By the way, this 1,3-bis-phosphoglycerate has a relative, and that relative is 2,3-bis-phosphoglycerate. I pray that you remember 2,3-bis-phosphoglycerate from our lectures on hemoglobin, because that is, you may remember, right on cadet, CO2 acid, diphosphoglycerate. Diphosphoglycerate is this molecule here, 2,3-bis-phosphoglycerate. So you can imagine in a red blood cell, this can get converted potentially to 2,3-bis-phosphoglycerate. You have a way of sort of linking glycolytic flux to hemoglobin delivery of oxygen to the tissues. So from 1,3-bisphosphoglycerate, we can potentially shunt this to 2,3-bisphosphoglycerate, which then goes and regulates hemoglobin. All right, so here are the phosphates phosphorylating ADP can make ATP. Phosphoglycerate kinase has a 3D structure very similar to hexokinase. Induced fit, Pac-Man. Okay, we literally call it Pac-Man in terms of sort of encapsulates that induced fit model of a substrate binding to an enzyme. Right, reaction 8 is a mutase, phosphoglycerate mutase. We're going to take that 3-phosphoglycerate and convert it to 2-phosphoglycerate. Okay, so the phosphate switches from 3 carbon to the number 2 carbon. Okay, the active site here is not a histidine, but a phosphorylated histidine. So how do we do that switch? It's a phosphorylated histidine. Phosphohistidine is the active site. So here's our substrate 3-PG, 3-phosphoglycerate, and here is that histidine. That histidine is phosphorylated. So we're going to actually produce an intermediary here that is diphosphorylated. Okay, the diphosphorylated intermediary sometimes leaks out. If it leaks out, it does leak out as 2,3-bisphosphoglycerate, which hopefully you now know is part of binding oxygen, regulating oxygen binding to hemoglobin. But if this intermediary doesn't leak out, what happens is just a phosphate exchange. So here is the phosphorylated active site. Here is our 3-phosphoglycerate. Here we produce that 2,3-bisphosphoglycerate. And then, so this phosphate here goes to phosphorylate the histidine. The 2-phosphoglycerate comes out. All right, so basically it's a phosphate exchange on the histidine active site residue. All right, slide here on 2,3-bisphosphoglycerate. Here's our first shunting pathway in. So this is our first shunting reaction, and it's basically taking glycolysis, which is what we were studying here downstream. But at this step right here at 1,3-bisphosphoglycerate, some of that can be diverted away, and that's called shunting. And it can be diverted away into this enzyme here, bisphosphoglycerate mutase, which converts it to 2,3-bisphosphoglycerate. So obviously we know from the hemoglobin lecture, 2,3-bisphosphoglycerate can bind to hemoglobin and sort of increase oxygen delivery. It shifts the hemoglobin from the R state to the T state, and the T state actually releases oxygen to the tissues. And then that 2,3-bisphosphoglycerate via another enzyme, the phosphatase, can regenerate 3-phosphoglycerate, which then can then proceed through glycolysis. So literally you have a metabolic detour. This is very critical in a red blood cell, where we have an intimate linkage between binding of oxygen to hemoglobin, that affinity, and also glycolytic flux. So the glycolytic flux is very high, and that is glycolysis is up and running with full throttle. Some of that 1,3-bisphosphoglycerate can be shunted off into 2,3-bisphosphoglycerate, which then goes into binding of hemoglobin and shifting that hemoglobin curve towards the more T conformation. Again, the T conformation is the oxygen delivery state of hemoglobin. But you can also take via this enzyme and have it shunt back into 3-phosphoglycerate and then continue glycolysis here. So shunting is an extremely valuable asset in metabolism. It's one of the reasons why we tend to split all these complicated anabolic and catabolic pathways into all of these multi-step processes. Shunting of 1,3-bisphosphoglycerate to 2,3-bisphosphoglycerate, very important in the cell that is the red blood cell. All right, so reaction 9 is 2-phosphoglycerate gets converted to phosphoenol pyruvate. Check out your arrows here, back and forth. The enzyme that does this is called Enolase, and what this basically does is it takes this form here and converts it to the enol form. If you remove this hydrogen and this hydroxyl group, it becomes water, and what happens is that this becomes a double bond. It's a dehydration reaction, essentially. You remove the water or remove the hydrogen and hydroxyl group in the form of water, and you get this very high energy molecule known as phosphorenol pyruvate. So phosphorenol pyruvate, by its very essence, is not a very stable molecule. It's highly charged, it's highly primed, it has what we call a high group transfer potential, meaning if this phosphate group leaves, you actually generate a lower, more stable energetic form of the molecule. So this PEP, phosphorenol pyruvate, it's really fully charged and fully primed. And that leads us to the terminal step of glycolysis. That's phosphorenol pyruvate, is a high energy molecule. It will phosphorylate ADP, and it's going to make ATP here. And then that phosphorenol pyruvate, by virtue of liberating that phosphate, becomes the molecule here, pyruvate. So remember here that we're actually producing 2 molecules of pyruvate. Everything gets multiplied by 2. From reaction 6 onward, and we also make 2 ATP here. Right? So from reaction 6 to reaction 10, we actually make 4 ATP. Right? Where did we make those 4 ATPs? Well, 2 of them come from phosphoglycerate kinase, and 2 of them come from this reaction here, pyruvate kinase. So 2 plus 2 is 4, but if you remember the hexokinase step, and then the phosphofructokinase step, enzyme 1 and enzyme 3, we actually had to utilize 2 ATPs. So the net effect of glycolysis is to generate 2 ATPs. 4 ATPs from reaction 6 to 10, we have to subtract 2 of them that were expended in reaction 1 and reaction 3. So we have 2 ATPs here, and we have 2 NADHs. The NADHs come from the enzyme glyceraldehyde 3-phosphate dehydrogenase. Pyruvate kinase, associated with chronic hemolytic anemia, so that enzyme that's mutated or in a dysfunctional form leads to hemolytic anemia, especially in red blood cells. A lot of these glycolytic enzymes, as mentioned before, when they're not in their native state and in their red blood cell, causes a lot of different types of anemias, the inability to produce mature red blood cells. Free energy of this reaction, very, very exergonic, substrate level phosphorylation, and we're making ATPs. So the mechanism of pyruvate kinase is basically nucleophilic attack. The ADP does nucleophilic attack of the phosphate. The phosphate is the leaving group, very high energy molecule, the phosphorenol pyruvate. And then after ATP generation, we have the generation of pyruvate. So the enol pyruvate tautomerizes into pyruvate. Right? And so this is the enol form, this is the keto form, okay, pyruvate. So we've actually completed glycolysis. We have made 2 molecules of pyruvate. So not something that's important for glycolysis or this specific lecture, but I just want to tell you that if we want to do the gluconeogenic route, if we want to go to the biosynthetic route, what we actually have to do is we take a molecule known as oxaloacetate. Oxaloacetate gets converted to phosphorenol pyruvate, and phosphorenol pyruvate goes straight up into the gluconeogenic pathway. Oxaloacetate is that molecule. Oxaloacetate actually comes from the TCA cycle. So everything is connected in metabolism. So here we have a metabolite from the TCA cycle, oxaloacetate, that can be converted to phosphorenol pyruvate. That's a 2-step reaction. And then from phosphorenol pyruvate, it's just all the way going back up to the gluconeogenic route. All right, so the metabolic fate of pyruvate, I will have a separate lecture on this because it's worth going over. But I just want to end this lecture with 2 things. Number 1 is what do we do with the pyruvate? Okay, what's next? Well, if you are a aerobic eukaryote, or if you are a highly advanced or evolved mammal, you can take that pyruvate and have it undergo the citric acid cycle. The citric acid cycle occurs exclusively in the mitochondrial matrix, and it produces a fully metabolized form of the pyruvate. It's a 8-step process, and it's a bicycle. So, it's a cycle. So it just runs on a cyclic step. What you do is if you take the citric acid cycle and combine it with the urea cycle, you actually get what's called the Krebs bicycle, but that's not something we will study in this class. So you can make it go through the Krebs cycle, also known as the citric acid cycle, and you'll get a ton of NADHs, and you'll also get some NAD pluses, the oxidized form. Well, that is for an aerobic organism. What if you are a bacteria? If you're a bacteria, or if you're a muscle cell that's undergoing high respiration, that pyruvate can actually be generated in one step to lactate. So pyruvic acid to lactic acid is a one-step enzymatic reaction. Lactate dehydrogenase does that. A lot of lactobacillus bacteria do that. And what happens is that you will oxidize that NADH to NAD+. Other bacteria, some other microorganisms take that pyruvate and in a 2-step process convert it to ethanol, and you will produce NAD+ from that. So why am I telling you this? Forget about the aerobes for a minute. Forget about the mammals. Let's focus on the bacteria. What's the purpose of taking pyruvate, converting it to lactate, or taking pyruvate and converting it to CO2 and ethanol? What I want to tell you is the NAD+. See, this NAD+ goes back into this step of glycolysis. It goes back into the glyceraldehyde 3-phosphate dehydrogenase reaction. So for simple bacteria, you need to get that NAD+ back to allow this enzymatic reaction of glycolysis to continue. This is the reaction 6, or enzyme 6 of glycolysis, without which glycolysis cannot work. So for highly technologically evolved or metabolically evolved species like humans or eukaryotes or aerobes, it's not a problem. But for anaerobic bacteria, fermentative bacteria, muscle cells that are really respiring at an active rate, you need to regenerate NAD+, the oxidized form, so that it can replenish the glyceraldehyde 3-phosphate reaction, glyceraldehyde 3-phosphate dehydrogenase reaction in glycolysis. Okay, definitely very important for bacteria. So that's a critical step for those microorganisms. So where will lactate go? Or excuse me, where will pyruvate go? Several fates for this, and we have a separate dedicated lecture for that. But pyruvate can go to lactate. Pyruvate can go to ethanol. That's a 2-step process, so you can actually genetically engineer E. coli to make beer or ethanol. That pyruvate can also go to oxaloacetate, which is part of the TCA cycle. All right, and what I've also mentioned to you is that oxaloacetate can actually, in a 2-step process, get converted back to phosphoenol pyruvate. That's a 2-step pathway. Oxaloacetate can also be converted to aspartate, so that's amino acid metabolism. This pyruvate can actually get converted to the amino acid alanine, that's amino acid metabolism. 3-phosphoglycerate in glycolysis, that's a glycolytic precursor, can get converted to the amino acid serine. So we can actually funnel some of these metabolisms to amino acids. And so that's a whole other branching. Also, that pyruvate can go to acetyl-CoA. And when it goes to acetyl-CoA, it does it through this multi-enzyme complex called pyruvate dehydrogenase multi-enzyme complex. So this is what's called a superenzyme. It consists of E1, E2, E3, and that's a fully committed step to the TCA cycle, that enzyme pyruvate dehydrogenase complex. So there's definitely several fates for pyruvate. And think about this for 1 second. Okay, if you have one, or if you ingest one glucose molecule, those carbons from that glucose can go into making lactate. Okay, For a bacteria, that may be important. Those carbons from that glucose can go into making ethanol. Those carbons can go into, from glucose, can go into pyruvate, which can go to oxaloacetate, which goes into the TCA cycle. Those carbons from pyruvate, from glucose, which goes to pyruvate, can go into oxaloacetate, which can get converted to the amino acid aspartate. So from glucose, those carbons can go into aspartate, that amino acid theoretically. From glucose, from glycolysis, those carbons can go to pyruvate. From pyruvate, we can go to alanine. So alanine can eventually come from glucose. And you can have this game go on and on and on. So that's the beauty of metabolism, tracking where all of the atoms goes.